But you all did a great job because every worksheet that I saw had the answers in the boxes. So you figured it out, and I appreciate that more than you know. All right, got the YouTube set up. All right, so while we're waiting, also you can go to Canvas. We're going to stay in Canvas for most of the lesson today. But again, if you have not submitted your other worksheet while we're waiting on folks, please use this time to do that. But if you have already done that, please go to Canvas, look over this page. We're going to go through all of the activities and videos and finish up this module. And again, I appreciate those of you who are always on time for class. All right, I'm gonna give it another few minutes. Because at this point, folks are officially 5 minutes late, but only half of you are here. All right, so I'm going to just go back a little bit. Like I said, they're late. I will go ahead and get started. All right, so last class we were looking at um, DNA replication. We did the gizmo where we made a copy of the DNA. And one of the reasons we make copies of the DNA that it's important for it to replicate is that the DNA does not leave the nucleus. While it has all of the information that codes for what the cell should do, what it should look like, how to function, that information doesn't leave the nucleus. So in order to get to the rest of the cell organelles, for them to know what to do and how to do it, and even make new cell parts, that information has to get out of the nucleus and make its way to where these proteins can be made so that they can, that the rest of the cell can carry out its functions. So what we do with the copy is like taking your instructions and going to the machine. Somebody tells you, and this is famous with testing situations, you can read the test booklet, but it cannot leave the office. So sometimes you make copies of the original stays in the office, and you may go to the Xerox machine and you make a copy of it. And the copy can leave the office. And then you can take it to the workers or the people who have to build whatever is in the instructions, and the originals stay in the office. So that's what DNA, the concept of DNA replication is about. We want to make a copy that can go outside to our ribosomes where the information can be read, and we can go ahead and make the protein that is being instructed by the DNA. All right, so if you're looking up here at the first line, it says that our ultimate goal for the DNA is to make proteins. Everything in your body is made of proteins. All the structures are made of proteins. So your protein and water and some other vitamins and minerals and things like that floating about, but you're mostly protein and water. Okay, so we have to learn how that process works. It's an assembly line, so we have to know how the assembly line works. We talked about in the last session that there are 3 different types of RNA. So the person who reads the RNA is the messenger RNA. Let me see if I can go back to that too before we do this. All right, so everybody should be in Canvas. We're going to be in Canvas today. Everybody should be in Canvas. All right, this is where I wanted to look at. Okay, so we have double-stranded DNA, and then again, you're going to have to remember the difference between DNA and RNA. So again, I'm backing way up. All right, so RNA has a different kind of sugar on it. They have the same structure. There's still a sugar, a phosphate group, and a base. So that unit is still called a nucleotide on both of them. But the difference between them is that RNA is single-stranded. Okay, RNA has uracil instead of thymine. And RNA can leave the nucleus. There are 3 kinds of RNA. The sugar, sorry, the other one was the kind of sugar that's in RNA is ribose sugar, whereas in DNA it's deoxyribose. Okay, so you need to be familiar with what those differences are. All right, so our big difference, other big difference with the RNA is there's 3 different kinds, and there are 3 different kinds depending on what their job is. So the one that's in the nucleus that just copied the information, that's our first one, the messenger RNA. So you need, and it's abbreviated mRNA. So the messenger RNA receives the carbon copy. Okay, we just went to the Xerox machine. Everything that was on the DNA strand, we copied it, and now the messenger RNA has it. It can leave the nucleus with this information. All right, when it does that, it goes to the ribosome, and the ribosome is the factory. And you have an assembly line, and the ribosomal RNA is lined up to receive the message or the instructions from the messenger RNA. So we're going to look at first translation and transcription. Those are going to be our 2 vocabulary words today, because first you have to translate and then you have to read what you translated. Okay, so we're going to look at the difference between transcription and transcribing. All right, so again, the rRNA is in the ribosome. That's what the R stands for, ribosomal RNA. And it is going to be the type of RNA that reads the message that came from the nucleus. And then its job is to, okay, now I know what to do. I've got to go get all of the parts in order to make this protein. I've been instructed to make a protein. I need to gather all the parts and put them together to make this protein. So this is where our third RNA comes in, the transfer RNA, and it's so the T stands for transfer, and it's the one that actually brings all of our amino acids. So again, going way back to the beginning when we talked about macromolecules and we said the units of a protein macromolecule is an amino acid. So amino acids are long chains of, proteins are long chains of amino acids. I always like to think of it like a string of pearls. So you think of these little round things, because they typically represent amino acids as little circles. So when you link them together, they look like a string of pearls. So, and then once you finish that string of pearls, that is your protein. That is the protein that the DNA said to make. And then that protein can go off and do whatever it's instructed to do. Okay. So that's just a backtrack review of what we should know so far. All righty. All right, going forward to today. So let's come back to everything is slow today. All right, so we've already replicated our DNA. Now we need to know what to do with the information we just copied. All right, so that's what we did. We reviewed the forms. Remember the enzymes help make these activities happen. And uh, and we do this to maintain homeostasis. That was a word we had way back at the beginning. Homeostasis simply means to create a balance in your system. So we have to make proteins in order to keep our body in homeostasis. All right, if you look over here to the side where it says protein breakdown, again, reviewing back from what we did earlier, we started off by saying that the elements that goes into making our macromolecules are carbon, hydrogen, and oxygen. In the case of proteins, nitrogens are also included. Now I'm looking at this little circle over here to the right. All right, protein monomers, again, are amino acids, and the function of a protein um, is to build enzymes. So enzymes are a type of protein. I don't disagree with all of this. Um, Proteins are needed for you, like your skin is made of protein, your eyes are made of protein. A lot of your heart muscles are made of, all your muscles are made of protein. So that is the function, is for structure and covering and protection. So proteins have a lot of function. It can, as a last resort, provide you with energy. When your body doesn't have enough carbohydrates to provide it with energy, you can break down proteins to provide you with energy. Another type of protein we talked about earlier in the semester was enzymes are a type of protein. Hormones are a type of protein. So all of those things are being made in this little process that we're going to look at today. Okay. All right. Antibodies are types of proteins. All right. Okay, so make sure you summarize that macromolecule about a protein. All right, the two T's. So this is what I was talking about earlier, that we're going to have to recognize which one is going on at what time. Okay, it says imagine something as small as the DNA can carry out instructions in making new organisms. The secret is the DNA nitrogen bases. So those, remember, there are just 4 nitrogen bases. The order of the bases code for specific traits in each living thing. So again, We said even though there's 4 of them, it's like our alphabet has 26 letters. Depending on how we arrange the letters will depending on the word that we make. With the 4 nitrogen bases, depending on the arrangement that they're in, will determine which proteins are made. And then we're going to find out that there's only 20 types of proteins. Okay, so those 4 bases will make will code for, and we're going to keep using that word code for specific proteins. Okay, and then we're going to have a chart, so you're going to be able to look up and decide what protein are we making. So there's a lot to unwrap with this particular section. But again, we're going to use that term code for. Okay, the DNA codes for a particular type of protein. All right, so the 2 words that we're looking at are transcription and translation. All right, we're going to watch this little video.
Every activity carried out by living cells uses one or more proteins. How are proteins made? The process called protein synthesis begins in the nucleus of a cell within a chromosome made of DNA, which stores the genes or recipe cells use to make proteins. The process begins when the section of DNA that contains the plans for a protein is copied or transcribed.
All right, I'm going to stop it there so that you know that that's step number 1. Transcription happens first. And where does it happen? It happens in the nucleus. So we need to know when it happens and where it happens.
The 2 strands of DNA separate, and free nucleotides pair with their complementary DNA bases. The new molecule formed is called RNA, which contains the base uracil, U, rather than the thymine, T, of DNA. The RNA is set free, and the DNA ladder zips up again. The RNA strand is called messenger RNA, or mRNA for short, because it carries the genetic message out from the nucleus to a cell structure called a ribosome, where the protein is built. The average cell of a mammal contains some 10 million ribosomes. The messenger RNA joins to a ribosome. The bases along the mRNA are grouped into sets of 3, called codons. Each codon matches a complementary set of bases, called an anticodon. On another kind of RNA, transfer, or tRNA. Each tRNA also carries one of 20 amino acids. Here, the tRNA carries the anticodon UAC with the amino acid methionine. The ribosomes assemble the amino acids together into a chain called a polypeptide in the order that forms a particular protein. As the first tRNA floats away from the ribosome, it leaves behind its amino acid. The ribosome moves along the messenger RNA. Each transfer RNA brings along its specific amino acid. The amino acids are assembled one at a time in the right order. Protein synthesis ends when a special stop codon is reached. The string of amino acids has become a polypeptide chain. The ribosome then releases the mRNA and the polypeptide. The polypeptide chain folds into a 3-dimensional shape that is unique to each protein's structure and function.
All right, so I want to go back just a little bit to...
Assemble the amino RNA floats away from the ribosome.
All right, so I want to come back here. right, so we had transcription. It happened in the nucleus, and it is the messenger RNA making a copy. So it transcribes what the DNA wants it to know. Messenger RNA leaves, it comes to the ribosome. All right, so then you have this red strip. So again, you have to recognize these structures as well. You have this red strip that's in the ribosome. Blanked out there for a minute. So this big blob looking thing represents the ribosome. And then what she said too is each cell has millions of these. So this factory operation is going on all over the cell. And you're constantly making new proteins for healing and building and things of that nature. All right. The other thing I wanted you to notice is this second, she mentioned triplets. So even though we have this long chain of bases, our A, G, and U and C, with the messenger RNA only reads them 3 at a time. Okay, so we call that a triplet code. So every 3 bases represents 1 amino acid. So then it's telling you this little kind of yellowish structure is your transfer RNA connected to, and there's a bunch of them floating around. There's 20 different kinds of amino acids, so there's 20 different kinds of these transfer RNAs. Depending on what the triplet code is, that's the one that will match up. And it brings along with it its amino acid. So then she said that these amino acids will start linking. So it's reading the code up here at the top, but down here to the bottom, the amino acids are linking together. Okay, the other thing you needed to notice, so every time another set of 3 is read, another set of 3 is read, you're adding another amino acid. So now we've got 3 of them linked together. And when they finish, when it gets to the end of the line, there is something, but she said a stop. It tells it when to stop making that protein. Don't go get any more amino acids. We're going to stop here. That's the protein that we're trying to make is already made. Okay. All right. Once it's gotten to that part, like you said, the chain of amino acids is called polypeptides, and we used that term before, poly meaning many. And peptides are our protein amino acids. All right. Thing that she said at the end, when this thing is done, the transfer RNA goes away. It waits till it's time for it to do a job again. And we now have this chain of amino acids that are connected, and they go off and they're ready to be whatever structure they were created to be. So the transfer RNA delivers the amino acids, lets it go, and then it goes back and gets another one from floating around in the cell, and then is waiting to come back to the factory when it's needed. Okay? So that's how that process works. So our 2 words, transcription, that happens in the nucleus, that's reading the DNA. Translation happens in the ribosomes, and that's our RNA translating. That's like is a foreign language. We need somebody to tell me what it says. The RNA then translates what was told to it from the nucleus. Okay. All right. So know these 2. So the transcription process of copying the DNA code and then carrying the copy to the ribosome. Translation is, and I don't like when they use the word to describe a word, but translating the DNA message into amino acids, or changing the message, converting the message into instructions that say which amino acids do I have to go get to build this protein. All right, let's look at this one.
Step 1 of protein synthesis, transcription. It is the process of forming a short strand of mRNA from one gene on a long DNA strand. The mRNA strand serves as a disposable photocopy of the master DNA code for a gene locked in the vault, which is the nucleus. We wouldn't want to use our master code, the DNA, to make our protein recipes, right? We use a photocopy mRNA instead. In this stage, we are changing the script of one gene from DNA letters into RNA letters. Step 1 of transcription, initiation. The DNA double helix is separated by RNA polymerase to make a transcription bubble at the start of the gene desired. RNA polymerase begins matching complementary base pairs of RNA nucleotides to the DNA. Note that it matches U with A. RNA does not have thymine. Transcription Part 2. Elongation. The mRNA sequence elongates as RNA polymerase moves along the DNA. This occurs at a rate of 40 nucleotides per second. Transcription Part 3. Termination. RNA polymerase reaches a termination sequence and detaches from the DNA. The completed mRNA molecule is released. The result of transcription: a strand of mRNA that carries a copy of DNA, which are the instructions for making proteins. The mRNA has a start and stop codon. A codon is 3 nitrogen bases or letters on mRNA or DNA that carries the code for an amino acid. The mRNA now travels from the nucleus to a ribosome.
All right, I want to come back and look at that again. All right, so this is our strand of messenger RNA. And notice that it starts reading at the top, and it does it 3 bases at a time. So each of the 3 bases is called a codon. And it only wants to read, if you're coding for a particular gene like eye color, height, that sort of thing, it's only going to read that section for that information. So it knows when to start and it knows when to stop. Okay, so all of this information here is the codon information for making a particular gene. All right, so that was transcription. Now let's look at translation. So all we did was make a copy right now.
Step 2. Translation. The purpose of translation is to make amino acids using the instructions in the mRNA strand. Remember that amino acids have to be made first since they are the building blocks of proteins. When the mRNA strand gets to the ribosome, it is in the wrong language. The mRNA needs to be translated into protein language, amino acids. Step 1 of translation, initiation. mRNA is transported to the cytoplasm and attaches to a ribosome so that the start codon is read first. A tRNA with an anticodon that is complementary to the start codon arrives at the ribosome, donating its amino acid. The tRNA is recycled and is free to pick up another amino acid. Step 2 of translation, elongation. The ribosome moves along the mRNA strand, reading the next codon of the mRNA in order. A new tRNA with an anticodon that is complementary to the next codon arrives at the ribosome, donating its amino acid. The tRNA is carrying an amino acid that is specific to that codon. This process repeats for an mRNA codon. The chain of amino acids at the ribosomes grow as more codons are read. Step 3 of translation, termination. When the stop codon is reached, translation stops. The mRNA leaves the ribosome. The polypeptide chain will fold into its three-dimensional shape, be taken to the Golgi apparatus where it will be processed, perhaps joined with other polypeptide chains, and packaged to be delivered as a final protein product to wherever it is needed.
Okay. I'm going to let you all work on this one for a few minutes. I'm going to stop sharing because I need to do attendance, and then I'll come back and share again. All right, so work on this matching section, and then we'll look at making sure you comprehend what we just talked about. All right, so everybody should be doing that practice session, please. All right, so except for the DNA replication, all assignments have been graded. Progress reports went out this week, so some of you need to work on why you have all these zeros. Okay, so letters go out today, letting parents know that you got some work to do. Just clarification again on the extra credit assignment, because somebody asked that it's showing up as missing. Don't worry about the CBN, mitosis, and cell cycle activity. It has zero value in the grade book, So this does not affect your grade. So it might say missing, but that's just to differentiate those who did it and from those who didn't. If you did it, you got 10 extra points. Okay, so that will add up on you. Because of the way the grades are done as a total, you either got the extra points or you didn't, but you are not penalized if you did not do it because it does not add up to your total points. All right, so is that clear to everybody? Like I said, somebody asked me. I can't control how it doesn't show missing on my side. It just shows 0, but the 0 is not, but it has 0 value. That assignment has 0 value, so that 0 is okay. That's the only 0 that's okay. If any other 0, you need to figure out what you want to do with that. Also, things, again, Canvas knows when the due date was, and it will calculate. It will calculate so many tenths, or I think like a quarter of a point for every day that it's late. So if I don't grade it as being 100, it's not going to get 100. And at some point, it's going to calculate as a 0 anyway. Okay, the only way it won't end up being a 0 is if I take the late off, and then I'll allow you to have half credit for the assignment that was late. So I'm not saying that you shouldn't do it. So it's always better to do it than to not do it. It's better to have half credit than no credit. So, but what I also don't want you to do is wait till next month when December rolls around and you decide that there's 6 assignments that you need to do all at one time so that you don't fail. Okay, so please don't do that because there is a date that I will stop grading late assignments. So please be aware of that. So if you're going to do late assignments, do them now so that we're not all piled up next month before exams. All right, that being said, so like I said, I'm up to date with everything except for the replication activity that you need to submit, and there's about 6 of you, 5 of you who still need to submit that. Okay, that's due today, so please be aware that after today, that's late. All right, so let's get back to what you were looking at in the module. Mhm. Okay, so you were supposed to be working on trying to match up what the 3 different types of RNA have to do. All right, so we need to know who copies the DNA and carries the copy to the ribosome. This looks like they're already in order. Translates the message into an amino acid. They are in order, as the amino acids are attached to a transfer RNA, and where the proteins are formed, they are formed in the ribosome. Okay. All right, so those were already in order. So if you haven't gone to the second page, here we go. Now you have to put this in order. So hopefully you did that. So what is going to happen first? I guess we're going to drag that up here to number 1. All right, so our DNA, and this is what you did with the worksheet that you submitted. Our DNA double helix has to separate. And we had the RNA polymerase, which is the enzyme that causes it to unzip. And it makes a transcription bubble. So that's number 1. And you might want to write these down on a separate sheet of paper, even though you have it in Canvas, write it down. once you get them in the correct order. All right, so now um, we have a transcription bubble. All right, so the next thing is it's got to the polymerase is that little thing that we had in the other movie. It's going to move along the line and it's going to read the DNA, and then they're going to bring matching. So as it reads it, it's going to bring matching complementary bases, meaning it's going to see that there's a T here. I need to go find an A. There's a C here. I need to go find a G. So this is what we mean by matching complementary bases. It travels from the nucleus to the cytosol. So, okay, that's not in the correct order. So first we need, okay, so we need to move the, there we go. All right, so after we get the complementary bases, it's going to reach a termination. And in other words, it's not reading the whole DNA, it's reading a section of the DNA. So there's going to be a place where it stops. And then once it stops making the messenger RNA, that messenger RNA can leave the nucleus and then it goes to the cytoplasm. All right, that better be right. No, why is that not right? Did it move it back? Okay, did it just change back? Uh-oh. Can I go back? Oh, it won't let me go back. That was right. All right, so that was transcription. Now we are translating. So we got to put this one in order. All right, hold on a second. I want to go back, but it won't let me go back. Okay. Okay, we want to get started. So we're in the ribosome and it has to line. So the ribosome is a structure. It's like the factory. The messenger RNA has to come to the factory. So they line up. All right, next. Actually, I guess this happens first. Okay, that it is transported through the cytoplasm and attaches to the ribosome. Then the ribosome can line up. Okay, we know this is last. And we're going to stop. Okay. These two sound the same. Let's see if it says it's correct. All right, so the messenger RNA left the nucleus, it travels through the cytoplasm, goes to the ribosome. Once in the ribosome, it's going to line up, create a template that needs to be read. So this is what we mean by translate. The message needs to be read, and it's going to start at the start codon, and it's going to read, and it's going to decide, I need this amino acid to be created because it's a triplet code. So it tells the transfer RNA, go get this amino acid. All right. As it goes through and reads the codon, each of the amino acids are brought to the ribosome by the transfer RNA. And these two are the ones I'm not sure. And then it's going to stop. It knows when to start. It knows when to stop. And then once it's finished, the chain of amino acids, which looks like a straight chain, then curls up and forms a three-dimensional molecule. See if this one is right. Yay. All right, so that one is correct. All right. So let's go back and look at why it didn't. All right, so this one it did correctly. Let's see what's wrong with the next one. Because I don't think that one was wrong, even though it told me I was wrong. I don't think it was wrong, but we'll see what it was saying that was wrong. Seems to be stuck. But in any case, hopefully you got yours correct. But you do need to know this little box. So this is a quiz box right here. Knowing which RNA does what it does when it gets to the ribosome, and what the transfer RNA is doing in order to assemble those amino acids. All right, so we need to know the difference between transcription and translation. What I want to do, because we don't have time this class, is to do an activity where you actually practice transcribing. But being as we only have a short period of time, well, actually, I can do that. All right. Take a look at this, because this is this little box over here. So go to the next, and it's not published. Okay, there we go. All right, mutations. So we're not so much concerned with the mutations as we are in this little box right here. We're going to learn how to read this box where it says messenger RNA codon table. And sometimes it's done as a table and sometimes it's done as a wheel. But this is how we determine which amino acids we're putting together. And notice on the table, it's done in sets of 3. And notice it's done in sets of 3 on the wheel. So it just depends on which one you like best. Okay. So okay, let me try something. But do read this, because this is we're going to look at using these two tables in order to construct a messenger RNA. All right, the gizmo. I want you to do this gizmo, but I need to make sure that it's going to work for you. Because the other day it didn't work. Okay. But I want you to practice. When we do this, I'm going to check with them as to why it didn't work for you. And it could be that the county license has expired, because you have to have a license to do this. All right, back over here. I don't know why it jumped out. Um, all right, so if necessary, I will do with this one like we did with the other one, but I want to start with it right now. Okay, so step 1, this is our DNA, and the first thing we have to do is to unzip it. So we're going to drag that over there, and we're going to unzip this DNA. Okay, so this first First one up here is TAG. So we need to have one that is an A. And then we need, and because we're making RNA, we're not going to have, RNAs don't have T's. So even though T is usually the complementary one for adenine, we're going to use U, okay? And then we're going to do. So just like the last time, we're going to have to make a complementary strand. Before we were making a duplicate of the DNA for the purpose of mitosis and meiosis so that we can make identical cells. Now we're not trying to make identical cells. We're trying to translate the information. So this is different to mitosis and meiosis. So our strand that is being copied is not exactly the same. Although it's the same information, it's just that this is how we know an RNA from a DNA is that there's going to be a U. RNAs can leave the nucleus. All of mitosis and meiosis, all of that's happening in the nucleus, and that information never leaves the nucleus. Now we're looking at something that's going to leave the nucleus. Shoo. Come on. And I'm sorry this is taking so long. Okay. So now you see this strand over here, and then you see what happened. What just happened? Somebody tell me what just happened. What did you just see happen? How would you describe what you see here? What did the DNA do? What did the RNA do? Anybody? And I don't want to have to do it again. But what did you see? What are you looking at now? How is this different from when I first started? What's separated? What is separated and what is not? All right, so Kendall said it's separated. What's separated? I hope y'all were paying attention because I'm going to ask these questions. These are quiz questions. What just happened? All right, this strand over here on the left, is that RNA or is it DNA? What is it? All right, Jacob said I made a messenger RNA strand. So yes, the strand that's single stranded, that it's not doubled, is the messenger RNA strand. And it left the nucleus and it's off to the side. What happened to the DNA that we were making it from? How would we describe what the DNA did as soon as it was finished making the RNA? What did the DNA do when it finished transcribing? All right, the two are separated, yes, but right now what I'm looking at is I'm looking at a single strand of RNA, and I'm looking at double-stranded DNA. And I wish I could reset this without make it go backwards without having to build it all over again. But what happened to this DNA as soon as that RNA was finished? Remember, we had a stop and we had a start and a stop. And as soon as it stopped making the RNA, what did the DNA do? I'm gonna ask this question again as a quiz question. All right, I'm gonna reset this and I'm gonna try to get you to, whoops, reset. Okay. All right. I took the enzyme and it started by, what did we call this process? What's it doing? What does the polymerase do to a DNA molecule? It causes it to what? To separate, or the other word that we call it, it unzips. Okay, so the two strands of DNA separate. All right. When I finished matching all of that up and we had our RNA, what did the DNA do when I was finished? So right now it's unzipped. When I was finished, what did it do? The RNA is made, yes, that's off to the side. But what did the DNA do when I was finished making the RNA? I wish I could do this fast. Thank you. It rezips. Jacob said it rezips. Thank you. So it zips back up. When it's finished transcribing, when it's finished giving its instructions, it zips back up. It goes back to being a closed up double-stranded DNA. Okay, so that's important. It unzips, it makes a strand of RNA. The RNA leaves, and then the DNA zips back up. Okay? All right. Okay, let me. I'm going to get you all to practice doing this. Do me a favor. Everybody try to, let me assign this to you first. This is not add to class. So I'm going to add this to your class. See if you can't, because I know the other day y'all couldn't get in. Try and see if you can get in today. This is, so we are going to go to Clever. And in Clever, you're going to go to Explore Learning. And let me know whether this is something that you can access. You still can't get in. Okay. I am going to email them and find out, because Mr. Phillips wasn't able to help me as to why you all, because y'all got in at the beginning. I've had classes work in this. I don't understand. And it has all the classes are registered. It is a county activity. So I don't know why all of a sudden you all cannot do this. But that being said, I will have the worksheets run off for you for next class. And I will, if necessary, I'll go through this and do this with you. And you all will just answer the questions and do the quiz. Okay. All righty. So we're going to let that go until next time. All right, but I do want you to read this right here so that you understand that there are 20 different kinds of amino acids. If there are 20 different kinds of amino acids, that means there's 20 different kinds of transfer RNAs, because each transfer RNA is coded to pick up one kind of amino acid. That's its job. It is assigned, it has a code for just that one particular amino acid. When the template is set, it goes and gets that one amino acid and brings it to the ribosome. All right. So there are 20 amino acids. There are 3 nucleotides in a codon. So you need to remember that. And I'm going to have a practice activity for you to do with that. Maybe we won't do the gizmo. We'll just have a practice activity where you just have to read the template and create an amino acid. So that's what we're going to do on Monday. Okay. So make sure you're reading this part here. All right. And I wish I could blow this up so that so we could work on this together. I'll have one that's bigger. All right. We get, let's see, how long is this? Not long. Okay.
Created using Powtoon.
All right. So, and like I said, we'll deal with mutations later. But the point is, if we don't read this thing correctly, we get a mutation, and the mutation says we read it wrong. Okay. So what you're going to have an activity where you're going to have to translate, and then if you don't translate it properly, you're going to give me a nonsense sentence, because the activity I'm going to give you, actually your amino acids are going to make a word sentence. If you don't translate it properly, your sentence will make no sense whatsoever. Okay. So that's what we're going to work. All right. So I'm going to let it stop there rather than try to force another activity. And so if you have no questions for me, like I said, some of you still need to submit your assignment. So please do that before you log off. Okay. All right. If you have no questions for me, have a good weekend.